This is Professor Doherty, and today we're going to be working on Lab 4, Molecular Movement and Rates of Diffusion. To begin with, let's go ahead and do a review of some terminology before we get started with this lab. First, this lab is heavily dependent on diffusion. Diffusion is the passive movement of particles, and that includes atoms, ions, or molecules, and all of those I would throw in are solutes, from a region of high concentration to regions of lower concentration. And this will continue until equilibrium is reached. So let's do a quick example here. Imagine that we have two beakers that are connected together through a tube, and that tube has a block in the middle of it. One of the beakers, we're going to put in a number of solutes, and this is going to make the concentration of solutes in that beaker high, and on the other side we are not going to put in any solutes, and that will make the concentration of the fluid in that beaker low. When we remove the blockage, what's going to happen is the solutes from the high concentration area are going to move to the low concentration area, and they're going to do this through a process called diffusion. And they will do this until the two sides come into equilibrium. Now, why does this happen? This happens because of molecular movement, which you can also think of as molecular vibration. What affects this? There's a number of things that affect this molecular movement. The first one is temperature. High temperatures cause more molecular motion or more molecular vibration. Think of it this way: if you put your hand on a hot stove, what you're actually feeling as heat is molecular vibration. Now, the second thing is the size of the molecules. Larger molecules move slower through their mediums. So a very large molecule is going to take a long time to move in comparison to a much smaller molecule. Next is concentration gradient. So in other words, the difference in solute concentration between the high and low areas. So if you have a very large amount of solute on one side and none on the other, then you're going to have a much faster diffusion rate. And then finally, distance. It takes longer for molecules to move over a longer distance. In this lab, we'll be looking at how size of a molecule and temperature of a solution affect the diffusion rates on molecules. Now, as a reminder, there's a couple other terms that you should know, including osmosis. Osmosis is the movement of water molecules, and typically this is going to be our solvent. You should also recall that water moves because it is attracted to solutes. So we use the term water chases solutes, and this is how osmosis generally occurs. Some other terms that you should know include dialysis, and dialysis is a little more complex. It's the diffusion of a solute across a semipermeable membrane. Now, you should understand that a semipermeable membrane is a non-living membrane. Its permeability is based strictly on pore size, and there are no active or passive transporters. Molecules smaller than the pore size will penetrate through the membrane, and ones that are larger will be blocked. So here's an example of this. So here we have a membrane, and here we have a large molecule. You can see the size of the holes in the membrane. The large molecule cannot pass through that small of a hole. If we have small molecules, they can penetrate through here, through this membrane. This is a semi-permeable membrane. Now, another reference that you should understand is selectively permeable membranes. Living membranes, for instance, like your plasma membranes of your cells, are selectively permeable. You can read about this in the boxed area on page 2 of your lab manual for the details on these living membranes. Now let's take a look at today's experiments. Today we will have two different experiments. The first one starts at the top of page 3 of your lab handout. This experiment will explore how temperature affects diffusion rate. We will fill 3 beakers with 200 mils of water each, then heat one of the beakers, leave the second beaker at room temperature, and we'll cool the third beaker. We will then add 3 drops of a dye to each beaker and record how long it takes until the dye diffuses to equilibrium within the water. To do this, we will simply watch the dye until it appears to be equally distributed within the water and then record the time that it took for that distribution to take place. Before we get started with this experiment, I would like you to look at question 1 at the top of page 3 and develop a hypothesis. We will then begin the experiment. Now let's begin our first experiment. Here we have our 3 beakers. This one has ice water, here is our room temperature water, and here is our hot water. You should record the temperature of each beaker's water in your chart on page 3 of your handout. Here are the numbers you should record. The hot water, or body temperature water, was 40 degrees Celsius. The room temperature water was 23 degrees Celsius, and the ice water was 5 degrees Celsius. Next, I added 3 drops of yellow dye to each beaker, started my timer, and then waited to see which beaker the dye diffused into fastest. Here are the results, which you should also record on page 3 of your handout. The hot water took 62 seconds for full diffusion. The room temperature beaker took 4 minutes, and the ice water beaker took 20 minutes for full diffusion. So from our experimental results, we can see that the higher the water temperature, the faster the dye diffused into the solvent. This matches what we would expect from the information that we went over at the beginning of this lab. Now, this is a relatively easy experiment, so let's go ahead and move on to experiment number 2. So now let's begin our work on part 2 of lab 4. In this part of the experiment, what we're going to be doing is looking at diffusion based on the size of a molecule. Now, for our purposes, the size of a molecule is equivalent to its molecular weight. So in order to determine the size of a molecule, we have to be able to determine its molecular weight. So let's go ahead and practice that. If you look at page 1 of your lab handout, in the middle of the page, you'll see that there's a number 1 where it says calculate the molecular weight of the following substances. These are the 3 substances that we will be looking at in this experiment in order to try to determine how fast they diffuse based on their weight. So first step, let's calculate the molecular weight of potassium permanganate. Here I will help you and step you through the process of calculating the molecular weight for potassium permanganate. First of all, we need to see the atomic structure, and we can see this here. It is KMnO4, and these letters represent the atoms that are within this particular molecule. Let's start with potassium. We need to determine the molecular weight of potassium, so we need to pull up a periodic table. On the periodic table, we're going to look for potassium, and we can see that potassium has a molecular weight of approximately 39.1 atomic mass units. Now, there are many different ways that you can say atomic mass units. You may hear it called Daltons, or you may hear atomic weight. For our purposes, these are all the same thing. Also note that I'm rounding off these numbers to the nearest tenth. Your instructor may want you to use all of the numbers. Here I'm cutting it off. Alright, then we record this number. Next, we need to look up the atomic weight of manganese, which we can see here is 54.9 AMU. We then record this number back on our sheet. And finally, we look up the atomic mass for oxygen, which is approximately 16.0, and we then record this number as well. Now we also need to multiply the oxygen atomic mass by 4 because there are 4 oxygen molecules in potassium permanganate as denoted by this number here. We then add all of these numbers together to get the molecular weight for potassium permanganate. Once again, I want to emphasize that the molecular weight of a molecule closely mirrors its size, so we can use weight as a surrogate for the size of a molecule. Now I want you to pause this video and calculate the molecular weights for Congo red and methyl orange on your own. Welcome back. Now check your calculations against my final numbers for Congo red and methyl orange. If your numbers don't match mine, then pause this video and go back and look for errors in your calculations. You should now be able to answer questions 2 through 5 on pages 1 and 2 on your own. If you haven't already done this, you can go ahead and pause this video here. Now we can get started with an overview of our second experiment. The procedure for this experiment begins at the top of page 4. To start with, we will procure 3 vials that are partially filled with agar. For those of you unfamiliar with agar, it is a gelatin-like substance, and we will be using the agar as a medium for our solutions of potassium permanganate, methyl orange, and Congo red to diffuse through. If this experiment works correctly, the molecules in the agar should resist the diffusion of the dye molecules based on their size. We will then place 3 drops of each dye on top of its own agar column and then wait 2 hours for diffusion to take place. After 2 hours, we will then measure with a ruler how far each dye was able to travel through its agar column. We will record these distances on the table on page 4 of your handout. Now that you understand the principles of this experiment, we can now actually run the experiment. Here we have our 3 dyes in dropper bottles along with 3 agar vials. I'm now adding 3 drops of each dye to their own agar vial. We then wait for 2 hours, and at the end of 2 hours our agar columns should look like this. Next, we measure how far each dye has moved in its vial. Here I'm starting with potassium permanganate, and it has diffused about 15 millimeters through the agar. You should record this as part of your data set on page 4. Next, I measured the distance methyl orange moved through its medium. I recorded this as approximately 9 millimeters. This is also part of your data set on page 4. And then I finally measured the diffusion of Congo red. Although the angle of the camera is a bit awkward here, the result is about 5 millimeters, which you should record. Also note that there is some dye that is leaked between the agar and the glass of the vial. This sometimes happens due to the expansion and contraction of the agar within the vial and can therefore be ignored as a known experimental anomaly. You should now have all the information that you need in order to answer the remaining questions on page 4 and page 5. Please check with your instructor for the correct answers to these questions if you have any difficulty. And with that, that concludes lab number 4.